finished on Tuesday. We finished talking about proteins, I think. So we walked over the water, right? Some water properties, pH, properties of carbohydrates, and proteins. The life molecules, lipids. We also talked about lipids. We did talk about proteins. So the last biological macromolecule that we need to recall some information about is nucleic acids. There are two types, right? DNA and RNA. DNA, which is represented here on the left, consists of two strands that form double helix. This molecule always fascinates me because, as I mentioned, in my opinion, it's probably the biggest advancement in science in 20th century, one of the biggest in the whole history of science. It also gave the birth to a science called molecular biology, and we actually know the date. That's kind of a rare thing for science. April 1st, 1953, the paper about structure of double helix was published. So it consists of 4 nucleotides. Don't mix up nucleotides in the DNA or RNA with amino acids in the protein. Just remember those are two completely different building blocks. So nucleotides, we'll talk about structure of nucleotide more later. So we have 4 nucleotides. There's 4 nucleotides, A, T, G, and C in the DNA, and A, G, U, and C in the RNA form strands. What is so unique about them? Pair how? Good. Excellent. And then A, U, G, C, right? So that means that they are complementary, which means that each strands carry the same, they contain the same information, and one strain can be restored based on another and vice versa. That makes sense? That actually provides like double storage, some protection against the single strand breaks, mutations. A lot of features of the living cell arise from that. Okay, the main function of DNA, of course, is storage of information, genetic information. If we will actually look at DNA, and I honestly try to think of any other DNA function, and I cannot. It's only storage. Of course, it participates in expression of that information as a storage. And I like that analogy. A lot of my analogies suck, so don't hesitate to tell me that. But this one really explains the idea. Imagine that you have a family cookbook. Gigantic, like twice the size of that notebook. Old one. You inherited it from your grandmother with all the recipes that your family was cooking all those years. Handwritten, typed on the typewriter, printed on the printer, all kinds of recipes. Only apple pie, you have like 15 recipes of apple pie. Seriously, like thousands of them. That's your DNA. Now, when you cook dinner for someone, you don't cook 15 different apple pies, right? Or 37 different beef stews. You pick one. One salad, one entree, one dessert, something like that. Okay? Now, if you have 1,000 recipes in that cookbook, are you going to hold the entire cookbook, which is your family treasure, to the kitchen? Probably not. I wouldn't. What I would do, I would take my phone. Well, now it's phone. Before, I would just write down the recipe that I want to cook on a piece of paper, right? Bring that piece of paper in the kitchen, not worrying about staining it or spoiling it or burning it, for God's sakes. and cook. So that piece of paper that I take into the kitchen, or photo of the pages on my phone, is the RNA. It conveys information to proteins, which are your dinner. Does that make sense? And that actually explains why, and it's a transition to the next topic we'll get to next, why not all the genes are expressed all the time. Because you don't cook dinner consisting of thousands and thousands of entrees. You cook only what you want or what you need at a certain point. Okay. Now, important thing is that when DNA is copied, new DNA molecules presumably carry the same information, right? That's an ideal world. Does that happen in real life? Exactly the same. Why? Mutations, right? Make sense? Mutations can come from mistakes while you copy. Mutations can come from damages to DNA. Actually, copying mutations, one of the examples that I really like, ever heard about the Bible comparison, that for the rich person, it is harder to get into heavens than for a camel to go through the needle thing. Ever heard that comparison before? Well, it's in the Bible, that for a rich person, it's harder to get in heavens than for camel, for some reason camel, to get through that little opening in the needle. And people were wondering, why camel? Turns out, the word camel and the word rope in Greek were very similar. So when they were writing it down, when they were copying the Bible, they made a mistake and translated. They thought it wasn't rope, they thought it was camel. So that kind of mistakes happens when DNA is transcribed, DNA is replicated. Okay? Just one mistake and it may change everything. Now expression happens through the messenger RNA. RNA is not double-stranded. It's single-strand, one chain of nucleotides. However, as you will see, DNA folds pretty much like this wire. Technically speaking, it is straight, right? But I have hard times straightening it up. Does that make sense? Now, RNA is a short-lived molecule that stores specific genetic information and conveys it to ribosomes to produce proteins. Make sense? And now, all the traits that we have, the result of protein expression. You agree with me? Like, the easiest one is eye color. Which protein is responsible for eye color? Skin color? Melanin. It's the same, actually. Melanin is responsible for eye and skin color. People who have higher melanin expression have darker hair, brown eyes, darker skin. Lower, blue eyes, green eyes, gray eyes, lighter skin. Make sense? It's all about protein. And the transition that I was talking about, that not all genes are expressed, that difference is manifested in phenotype. So you have to understand the difference between genotype and phenotype. All of us in this room, do we have the same genotype? From almost the same, very few differences. We do have some genetic differences, absolutely, of course, but on the very basic levels, on the large scale, we all have the same phenotype, same genotype, same set of genes. Those genes are very, very, very similar, extremely similar, maybe absolutely identical. Okay, why we look so different then? Exactly. expression of the genes, right? It's how genes are expressed. That's what defines phenotype. Genotype, of course, underlies phenotype, but expression of genes is what defines phenotype. Does that make sense? See that statement? Expression of genes defines phenotype. Each and every time. Is that good? Now if you think about that, look at us growing. Changes everything, right? Well, we're not talking about big eukaryotes like us. We talk about bacteria mostly, and protozoa. Do they have the same? Of course. So this is your basic scheme of gene expression. DNA is transcribed into RNA. RNA is translated into protein. This is often called the central dogma of molecular biology. It's kind of interesting because this whole sequence was initially proposed by James Watson, together with the guy that one of the guys that discovered the DNA structure. And later in his interviews, he said, "I never wanted it to be a dogma, but somebody coined that. Oh, that's now a new dogma of molecular biology, and so it goes." It's not even entirely true. Some other organisms use slightly different ways of expressing genetic information, but this is a very fundamental feature here that in one or another form will be seen in every living organism. Okay? Now, what defines genetic expression? What defines expression of genes? Environment. Say again? Well, phenotype is what comes out of the expression, but what is responsible for the way we look? Environment. It doesn't mean that if I will keep you underwater long enough, you will grow gills and scales and learn how to swim underwater. It's just too short of a time. It's a longer exposure to environment for us that defines phenotype. Okay? Because what is the generation time for humans? Huh? 25. Yeah, 20, 25, you know. It takes a hell of a time to get through like 3-4 generations. What about bacteria? Any idea? Wild guesses, huh? Depending on shortest in the lab recorded for Campylobacter jejuni was 13 minutes. So in 13 minutes, bacteria double. Yeah, exactly. Well, there are some that suggested to have doubling time of years. That's a different story. Usually it's very, very fast, right? What does that mean? It means, change quick. Yes, microorganisms respond to changes in environment quick. And when we talk about genetics, when we talk about changes, when we talk about evolution, we must not talk about one bacteria. We must talk about population. That makes sense? You have a million cells, and you start to grow them not at 37, but at 42. What's going to happen to most of the cells? They will die or they will stall, okay? But some proportion of those cells will have mutation, pretty much inevitably, that will give them the advantage and they will flourish at 42. And you will get temperature-sensitive mutant of bacteria. Does that make sense? See, they will adapt to environment quickly, okay? And they will produce different phenotypes depending on the environment. Does that make sense? That's an important feature here, okay? Now, because of that quick replication, what is the driving force of the change genetic change? Huh? Mutation. Well, in higher eukaryotes like us, what else? Huh? Survival is the pressure that drives the changes. Yes, sexual reproduction. Well, bacteria don't have sex in a traditional way. We'll talk about what is called bacterial sex, so-called conjugation, but it's not classic sexual reproduction to haploid cells exchange. Okay, so it's either mutations or some exchange of genetic information, and in this case, mutations, we're going to focus on them in this particular topic most. So mutations accumulate. They're going to be good and bad, right? They can be beneficial, they can be disadvantageous for the cells. And those changes allow bacteria to adapt. One of the best examples is resistance to antibiotics. Imagine that you have a population of bacteria and you put them in the medium that has incredibly high concentration of antibiotics. Antibiotics that are bactericidal, that are killing bacteria. Sorry. What's going to happen to all the cells? Like sky-high concentrations. They're all going to die. That's it. No adaptation. Seriously. You can do that. Another situation. You put all the bacteria in the medium and it has moderate or low levels of antibiotic. Most of them will die, but some of them will survive. And they will survive not by chance, but because they somehow resistant. They have that mutation that gives them the advantage. Make sense? And then if you will continue to grow them, they will create better and better population. In fact, we think about if you will do that in the lab and you start bacteria on the low antibiotic concentration, pretty much if we started today, by the end of the semester, we would have completely, say, doxycycline resistant strain of bacteria. Completely resistant. Because it gains resistance so quickly. That's your evolution. Okay? They evolve to adapt to anything. And when you think about a certain trait in the bacteria, you have to think about evolution. What's the point? Why? Why this trait? Okay? And this is the statement like in bold and caps. I'm not screaming at you here. Everything that we look at in this course will look through the lens of evolution. And it's kind of a repercussion of the statement that famous biologist Theodosius Dobzhansky said, that nothing in biology makes sense except in the light of evolution. Why I'm saying that? While we're at it, I want to ask you a question. What is the main purpose, life purpose of living things? Reproduce, that's it. Okay, so when you look at the microbe, Ebola virus, its biggest life achievement is not to kill, but to transmit to another host. Make sense? So when I have that respiratory tract infection, why? What is the evolutionary meaning for, let's say it's probably virus, although maybe different. Let's say I have rhinovirus, upper respiratory tract infection. I had runny nose, I'm coughing, I'm sneezing. From standpoint of virus, to reproduce and spread, why does it cause all those symptoms? Absolutely, so it can spread. Does that make sense? When I cough, I transmit it to other people. It's not because virus is evil and want to make me sick, no. Does that make sense? I love viruses because it gives you such a great perspective on evolutionary advantages. Ever heard about rabies? If I get rabies, can I transmit it to you? Unless I will really jump out of my pants, I will try to bite you a lot. People usually don't do that, actually. So for rabies, infecting human is death. Virus dies with the host. So it's absolutely dead end. Okay? That's accidental. So evolutionary, it's not meant to be. Does that make sense? Animals in the wild, the rabid animals, they survive for much longer time, and they bite. Oh my, they bite to spread the virus. If you think about infections that are most successful in transmission, any ideas? Infections that, virus infections that infect almost everyone. Huh? Say again? RSV, surprisingly not as many. Kids, yes, but adults not as many. Influenza, a lot, but in epidemics, yes, a lot. Very good. Does it cause like really bad symptoms? Not anymore, yes. Strep throat, not. I keep one infection in mind that up at a certain point infects about 60% of the world population. HPV, malaria much less than 60. HPV infects a lot, but it can be cleared. This one cannot. Chickenpox, yeah, chickenpox we now have vaccines. It used to be. Yes. Herpes is herpes, unlike love, is something that stays with you forever. It's a gift that keeps on giving, and it doesn't cause horrible symptoms. Okay? It may cause cold sore once in a year, maybe 2 years. Rarely. You're not going to die from it. So if you would look at the infections, the ones that cause the most severe disease, usually humans are not the primary host for them. Does that make sense? And on the other hand, the ones that infect almost entirely humans, they're very mild. You see, because they adapted to coexist with us. Because if cause dies, they die. Does that make sense? We always have to think about reproduction in case of why do they evolve this way. Okay? Any questions? So now we're going to switch to microscopy. And what I'm going to do, I'm going to grab, I don't have to grab the microscope. I have one over here. It's a little bit more fancy looking than the ones that we have right there in the cabinet. Actually, what we're going to do today, when we'll get those samples, we will learn, like hands-on learning about microscopy and how to stain things. Okay, so we're going to chat about it now, and then we're going to learn how to look at the microbes under the microscope. It's really important skill. So you have pretty much, what is the microscope? System of lenses. I always have problem with singular and plural in terms of lens. Lenses, you're correct. So it's system of lenses. You have ocular lens, eyepiece, and you have objective lens. Okay? Now, the microscope works at the principle of refraction. So it changes the direction of the light rays, and in some cases because light obviously doesn't go like this, it doesn't bend. So in order to direct it, you also have to have some reflective surfaces. Okay? So it's refraction and reflection of light. Okay? And what lenses do, all of them in the whole system, they increase the size of the image. That's one purpose of lenses. Lenses can be convex, Convex lenses will actually increase the size of the image. They will bring the object closer to you. When you use the magnifying glass, that's the convex lens. Okay, concave will actually move the object a little bit farther from you. Okay. We talked about, so that was the microscope invented by Leeuwenhoek, that Dutch guy that I told you about. It doesn't really look like anything modern, and that is the Hook microscope. So you see eyepiece, objective lens, table, no, stage, it says stage, the light source, all elements of the modern microscope. Hook actually observed the structure of the cork. He was the first to see cells. Well, dead cells. He never had that leap of logic implying that everything is built of cells, but he was the first to observe them. Now to kind of Leeuwenhoek, he was glass master. When he designed his microscope, what was the first thing he looked at? Any ideas? Nothing fancy, trust me. Quite an opposite. Huh? Oh, no. That was Hook. Leeuwenhoek looked at something, I'd say more, huh? Oh, you're too romantic. You're closer, yeah. Get more nasty. Huh? Poop. His own. And he discovered quite a lot of microbes there. He was just curious what it looks like under the microscope. He discovered quite a lot of them. Now, microscopes that you're going to work with are compound, which means they have multiple objective lenses. Okay, mine is different than yours in only one instance. You're going to have 4 different objective lenses with magnifications of 4, that's the scanning lens, 10 low power, 40 high power, and 100 oil immersion. Mine go 10, 20, 40, 100. So maximum magnification of this microscope is the same as magnification of yours. So it's compound. Light, obviously, yours and mine both use light. Light, compound, binocular. It can be a little tricky. Have you used microscope before? If you haven't, or you forgot how to do that, or you used not binocular, but monocular microscope. With binocular, you cannot do this. You cannot stick your eyes to the eyepieces. You have to be a little bit far apart. Okay? Like this. And then you will see one picture. Right? And it's parfocal, which is kind of an interesting thing. Focal means that when you change objective lenses from one to another, it remains in the relative focus. So you start with the lowest magnification. You find whatever you want to look at. You go to the next lens. And you just have to adjust your focus just a little. Make sense? You don't have to play focus all over again. Now, magnification is usually what we look at when we talk about microscopes. So how much bigger the object looks. Right? Magnification depends on 2 things. Magnification of your eyepiece and magnification of your objective piece. You multiply one by another. I'm going to tell you your eyepiece in all those microscopes. Eyepiece has magnification of 10. Okay, so what's going to be maximum magnification of the whole microscope that you can achieve? 1,000, 1,000, right? No, 10, a maximum objective is 100, 1,000. It's why I focus so much on that. if you happen to work in the clinical lab, or if you happen to look at the microscopic images, images will always give you the magnification under it. Like that sample of bacteria was taken at magnification of 400. It means it could be 10 eyepiece, 40 objective, or 20 eyepiece, 20 objective. Does that make sense? You don't have to think, oh, this guy, what kind of microscope did he had? Okay. Magnification doesn't equal resolution. You see the difference? Resolution is how sharp the object is. Right? You probably have seen it when you try to enlarge some part of the picture, like a photograph in the computer, and it becomes more and more grainy. It would always stuns me in movies about, crime scene investigation and stuff. They have really crappy picture of the license plate. They enlarge it 1,000 times and they can like see every letter on it. Okay, that's not going to happen. So resolution depends in microscope. It depends on mostly on the wavelength. So, well, wavelength, light, of course, more light surprisingly usually means less resolution, less contrast. Okay, we'll get to contrast. Resolution again depends on the wavelength. I'm not really good at physics, so the maximum I can tell you, look at the wavelength here. So you see the frequency is low, wavelength is big. Does that make sense? Low frequency, big wavelength. High frequency, shorter wavelength. The shorter wavelength, the better the resolution capacity of the microscope. Now, what is resolution? Resolution is ability to distinguish two closely located objects. Does that make sense? I usually, to myself, I explain it this way. So if we have two closely located objects, longer waves can just go around them. And shorter waves will bump into each of the objects. I know physicists will cringe about this explanation. But the idea is that the higher the frequency of the light, better resolution you're going to get. Okay? So what is the lowest frequency of the light? Which light has the lowest frequency, the visible color? Red. The highest? Blue. So we try to observe it closer to the blue part of the spectrum, if we can. UV light will have better, but we can't see it. Okay? Make sense so far? Contrast. Difference between dark and light parts of the image. Well, you can enhance contrast by staining your specimen. You can play with the light. There's a part of the microscope called iris diaphragm that adjusts the amount of light coming through the sample, so you can see your image better. It actually is very important part of the microscope. Little thing under the mechanics stage. Okay. I mentioned about the first microscopes and I'm not going to talk about it a lot. So that's the microscope that you're going to see. We've already talked about eyepiece, okay, objective lens. Well, objective lens are on the so-called revolving nose piece, so you can just rotate them. Okay, make sure that it clicks, because if your nose piece is stuck between two objective lens, or even just a little bit aside, you're not going to see an image. It has to be in place. Stage, mechanical. It means that you don't have to move it with your hands. You have knobs here, okay, to move it around. There are clips on the stage here. These clips will hold the slide. It's very convenient. You position the slide properly and you just move the stage around. You don't have to touch anything with your hands. There are two focus knobs. Sorry, two focus knobs. I hope you can see that. The bigger one is called coarse focus knob. It adjusts focus when you are on the lowest magnification objective, the scanning objective. Fine focus knob, the smaller one, here it is used on the higher magnifications when you are on the low power, high power, or oil immersion lens. Does that make sense? Why you do not want to use coarse focus knob on the higher magnification lenses? Huh? You can break the slide. Well, slide is okay, but you can break the lenses if you're really aggressive. Also, you move coarse focus and you just get completely out of the focal, you move focal point somewhere else, okay? So you lose focus completely. Light intensity on the side of the microscope you will see, so you don't burn your eyes. Condenser focuses the light on the specimen. Diaphragm regulates the amount of light that gets to the specimen from the lamp. Any questions? Now take a message from previous and next slide. So from here, understand the meaning of words like fraction, reflection, compound, binocular, parfocal. Okay, difference between magnification, resolution, and contrast. Good? Here, for each part of the microscope, understand why it is there. It's sometimes hard to explain why eyepiece is there. The best answer will look in the eyepiece. Just look in there, okay? It's just part of the lens system. That makes sense? Microscopy can be divided into two major categories: light and electron. There's some more sophisticated, like atomic force. Yeah, or tunnel microscope. It's tunnel scanning. It's really, really awesome stuff. But in microbiology, it is used sometimes. I mention it on pause just for the entertainment. So light microscopy, we're going to use bright field. Period. So this one. That's the typical microscopy that you have been using before. You will find in any other course that we teach here. Essentially, light goes through the specimen. Your sample should look, the bacteria, the cell that you look at, should look darker. Why something looks darker than the environment? It blocks light, it absorbs light, right? Now, have you looked ever at the water sample with a lot of protozoa floating around? Those protozoa, how they look like? Transparent or really dark? Yeah, they're usually transparent. That's a problem with those little creatures. They're mostly water. You see? Mostly water. Those of you who have taken before sometime will look at the tissue samples. Are they colored? Yeah, they are. They're colored. You know why? Because they were stained. Otherwise, you can hardly say anything about them. Because cells are mostly water. They don't absorb light that much. So in the bright field, if you would look just at the sample of bacteria, sorry, you can barely see it. Okay? So often we're going to stain. You see here, it's the staining of bacteria. It's bacillus and bacillus species that were stained to increase contrast between the cell and its background. Dark field. There's some light magic, physics magic, with the light actually coming on the sides of the diaphragm, so the light is directed somehow. Anyway, the background is dark and cells are lit up without staining. The advantage, you can fairly clearly see the structure of the shape of the cell, but internal structures sometimes are not as good. Does that make sense? Because there's just too little light. What is good, you can use it to observe live cells, because staining kills them inevitably. Okay, we're gonna try and observe those bacteria live. We may or may not see them. Probably will. You won't be able to tell a lot about them. But we don't have dark field on those microscopes. But if we would, we could try. Okay, and look at the dark field, the live samples. So you see Borrelia right here. It's spirochete, Borrelia burgdorferi. It's a spirochete that causes Lyme disease. I bet you've heard about it. Phase contrast. Next level of light trickery. They use some light interference to increase the contrast of the image. You can use it either with a live or stained specimen. Even with live specimen, the picture is pretty awesome. This microscope has the capacity, phase contrast. So when I will try to put some samples on the phase contrast, and you will have a chance to look how it looks like. Just those, I think this microscope is like $10,000 or $15,000. It's like a really expensive one. So phase contrast option is useful when more in the research rather than in the clinical settings. Okay. Differential DIC. That level of light trickery is beyond my comprehension. Pretty much the idea is that contrast can be so much enhanced that it looks almost like 3D image. Okay, that's max that you have to know. That's about it. Again, live specimen. Next, fluorescence microscopy. Now for fluorescence, you have to stain cells so they can start to fluoresce. What do you have to have in order for things to fluoresce? Yes, the black light, pretty much. Special light that will light on the, what you might call it, the specimen, okay, the stain, and stain will start to emit its own light. That's what we call fluorescence. So you have project UV on the specimen, specimen will light up. Now we have many different stains. Some of them will produce green light, some of them will produce red light, some of them will produce yellow light. You may have heard about little animals that fluoresce in the dark. I think now you can buy fish that glows green if you put in the black light. It's a green fluorescent protein. They created rabbit for sure, green glowing rabbit, mice, rats. I don't know if they sell them, but fish definitely, and it's fairly inexpensive. It's pretty neat. I'm waiting for creating red, green, and yellow and just put them all in the same aquarium. Now, what is advantage of fluorescent staining? Well, first of all, some cells, some bacteria are really small and it's hard to observe them using regular light microscopy. For example, mycoplasma, which is a common causative agent of pneumonia and your genital infections is really small micro. There is a special stain, fluorescent stain, that specifically stains mycoplasma. So what you can do, you can take a sample, stain for mycoplasma, and then light it up with the UV light. And mycoplasma cells will start to glow. You will not see internal structures. They will glow like little spots. Make sense? You can also attach fluorescent stain to antibody, which specifically recognizes a bacteria, a virus, a protozoa, a certain structure, okay? And then you can distinguish different cells one from another. It's all possible with a fluorescent light, okay? Confocal. So this image of confocal microscopy is not the best. Do you know the idea of MRI, how it works? I mean, not physics behind it, but what's the point? Why take an MRI? What does it produce? Or any type of tomography? Huh? Yeah, so, right, so it produces like a 3D image, right? So you can look at the slices. Same goes for confocal. It's pretty much a tomography. The fancy microscope, I frequently use the word fancy. Fancy in my lexicon means I have no idea about the mechanism, how it works. It's magic. So, and it pretty much costs a fortune. Confocals can cost up to, really good confocals, $250,000. So it's for instance, the institution I worked at before in Louisiana, they had, I think, one confocal microscope for the whole building. It was shared use. There were quite a lot of researchers. So it's not like you write the check from your grant and you buy it. It's not that simple. So confocal microscopes take slices at different focal planes, slices of the object. Does that make sense? They change focal plane and they make images, and then they combine those images, essentially producing a 3D image of what's going on. Does that make sense? That's your confocal microscopy. Okay? Quality can differ. 2-photon, I saw it. This 2-photon microscopy uses laser that goes into the specimen, and there are special stains. So it's combination of laser as a light source and fluorescent microscopy here. It can get into, as it says here, really thick specimens. So you see intestinal cells. What is the best part about 2-photon microscopy? You can add fluorescent stains that do not kill the cells and see movement of live cells. And if these stains can distinguish them, say you have stains for epithelial cells of the intestine and you have stain for E. coli, you can see how E. coli moves through the intestines. You can actually do it on the animal, that microscopy. That's also fancy. Electron microscopy. When you look at this microscope, what penetrates the specimen? Why do we see the specimen? What goes through the specimen? The light. What are the light particles? How do we call them? Photons. Okay? Instead of photons in electron microscopy, we use electrons. So we use electron beam, like a ray of electrons. It goes through the specimen. And there are two ways. First, if you use that electron beam to penetrate the specimen, like we're doing the light one, you're going to have transmission electron microscopy. You got to have a focal point. You got to have a slice. Does that make sense? So transmission electron microscopy gives you a plane of the specimen, a slice of the specimen. So if you take, I don't know, cell infected with Ebola virus. So this is Ebola virus. You have no idea, is it round shaped? Is it like this shaped? Is it rectangular? We don't really know because it's a slice. That makes sense? It's like a stick of salami. You cut it across. You look at it. You don't know how long it is, but you know that it's circular on the cross section. Does that make sense? That's transmission. And transmission and scanning can be easily separated based on the picture you look at. So this is how scanning look. Do we know what's inside those cells? No. But we know the shape. In my opinion, transmission, I would say it's ugly but useful. Scanning is gorgeous but not very much informative. Okay, we have Campylobacter here. Cool. Now what? But it produces really stunning images. And if you would Google scanning electron microscopy algae, good God, you won't believe that these creatures exist on this planet. They're like out of this world. So those are types of microscopy for which you should know basic idea. Believe me, it's basic. Again, I'm not going to ask you about that light trickery they use. Difference between light and bright field. Why we use bright field. What is the advantage of phase contrast? Well, increased contrast. Okay. Why DIC is, what is the difference of DIC? DIC gives you a 3D image. Fluorescent, you can color the samples. You can distinguish different types of cells. Confocal, 3D images. Do photon, live images, live cells in a thick specimen. That's pretty much it. Okay. Electron, know the difference between transmission and scanning. And I quite likely will provide you with an image on exam, and you will have to tell me what type of microscopy it is. It's going to be obvious, I promise. I mean, if you have an idea what they do, it's going to be obvious. Okay. I mentioned, just for entertainment, to say about scanning and atomic force. Scanning has a needle that goes over the surface of the specimen and measures the electrical current between the surface of the specimen and the needle, the distance like tenths of nanometer. So it can, for instance, see the structure of DNA molecule. And atomic force allows you to manipulate atoms. One of the first pictures using atomic force microscopy was produced by IBM company. Well, guess what? They used, I don't remember which chemical element, but they wrote IBM using atoms and made a picture. It blows my mind. Any questions about types of microscopy? Okay, next now it's time to talk about staining. So, how do we do staining? We usually, well, not usually, we actually don't do staining on just live specimens. We need to kill it first. Ugh. Looks awful. How do we kill it? So, first of all, what is the simplest way? to produce the microscopic specimen. It's wet mount. Have you done wet mount before? How do we do wet mount with the microbe? I don't have water here, so I'll just show you. So these are two slides stuck together. You don't have to use two, they just, for some reason, got glued. They're really, really nasty and dirty. Take a little drop of sterile water, it's right there. Put it on the surface of the slide. Then take this loop, sterilize it in the flame. Right there. Okay, sterilize it in the flame. Now, don't scoop your colonies, because the amount of cells that you're going to scoop is astronomical. Touch it. Sometimes when I cannot, like when I don't know if I'm going to pierce it through or do something else, I hold my hand and just touch it, and then you mix it in the drop. It's your wet mount. You're good. You cover it with a cover slip and go ahead. Look at it. That's ready to go. Okay? Now, what is fixation? Fixation is essentially killing the cells. What's the point? They're not moving. We're going to do heat fixation. You can do chemicals, but we're going to do heat fixation. Here it shows you fixing on the heating plate. We're going to do over the flame. Essentially, fixation over the flame. You have your wet mount. You have a flame, and that upper portion of the flame, you do 1, 2, 3. Okay, this is flame 1, 2, 3. Okay, yeah, that's it. It is fixed. Don't be stressed out. Don't be disappointed in yourself. For the first time, you will burn them. If you overheat bacteria, a lot of cells are going to explode. That's going to look really, really awful. And it's just not, you won't see nothing pretty much, just a bunch of crap. Okay? But you'll get used to it. Seriously. So you do this, and then when it is fixed, it is ready for staining. So staining provides color, increasing contrast, so you can see structures better. Make sense? Staining can be simple and differential. Simple means that all microbial cells are going to be stained the same. In your cabinets, you have boxes like that, various bottles. There's alcohol, don't drink it, in nature. There are plenty of stains. For instance, crystal violet and safranin are simple stains. All cells, all of them, whether it's Staphylococcus or Campylobacter, gram-positive or gram-negative, they're all going to be stained blue with crystal blue, crystal violet, red with safranin. Okay? That's simple. Differential allow you to distinguish microbes. Here you will find, somebody stole it from me, but in your boxes you will find the bottle called, gram iodine. So gram staining is one of the fundamental stains in microbiology. Okay? It allows you to distinguish between two big groups of bacteria, gram-positive, gram-negative, based on the color. It reflects actually a lot of bacterial features. It's not just red and blue. It actually is physiologically, biologically important. There are other differential stains, which we will talk about. Again, those stains allow you to tell different bacteria part, right? So this is example of differential stain, the Gram stain, okay? So simple stains, though, can be positive. So like this one. When bacteria is stained, again, going back to what I showed you, crystal violet is the positive stain, okay? It's usually the thing in the stain, that molecule of crystal violet is positively charged. So it will stick to mainly negatively charged proteins on the surface of the cell, okay? Positive, negative, okay? And it will stain the cell, not the background. Things like nigrosin are negative stains. They're usually negatively charged, okay, and then stain background, but not the cell. You can see it here. Okay, Bacillus megatherium on this image, image number 3, is stained with, I don't know if it's nigrosin or India ink. So this one, bacteria is not affected, but background is stained. So this is quite useful to determine the structure of the cell, the appearance of the cell. Does that make sense? Okay, it doesn't penetrate the cell. Well, differential, talk about them more, and you see the brief list that we're going to talk about. You can, again, see the Gram stain here. I actually did the color coding. So those purple ones, Gram-positive Staph aureus, and the red ones, Gram-negative E. coli. Any questions? So take home message. Understand why we stain. Understand why we fix. Understand what's the difference between simple and differential stains. And what's the difference between positive and negative simple stains. Okay? So here's the Gram stain. Oh, let's go back to differential, say a few more. So acid-fast stain. Acid-fast. Stains for mycobacteria. You've heard about at least 2 of them. Causative agents. Diseases associated. Tuberculosis. Right? Everybody heard about tuberculosis. And leprosy. Also caused by mycobacteria. Do not mix up mycobacteria and mycoplasma. They are not only different species or genuses, genera. They are completely different phyla of bacteria. Okay? Understood? Any questions? Couple more. Capsular. Stain that allows you to see the capsule around the microbe. And flagellar. Do we have understanding of what flagella is? Like a tail, right? What is the problem? Why can't we see flagella normally when we look at the bacteria under the microscope? It's very thin, right? And remember, I mentioned that when we talk about microscopy, resolution, the ability to see the elements of the specimen, is defined by the wavelength. We cannot go shorter wavelength than blue one. And flagellum is too small even for blue wavelength. So how would you think flagellar stain works? It's too thin. What do you have to do to make it visible? Yep, you make it thicker. You have special stain that sticks specifically to flagellum and makes it fat. And now you can see it. That make sense? Now, Gram stain. Probably not today. With Gram stain, I would wait till we get samples from the environment. By the way, you have 2 weeks to think what in the environment you will sample. It's always fun thing, really. Free your mind. Well, fecal samples can be interesting, but we may have problems collecting those. Okay. Mouth, nose, we got skin already. Floor, your purse, inside, outside. Your shoe, inside, outside. Technically, your pants, inside, outside. Not a lot of interesting things. It's going to be mostly your skin. Breakers, be stealth. They don't like it. Toilets, whatever you want. I mean, the goose poop outside. Seriously, anything. We can collect it. We'll plate it. We'll have colonies. And then, my plan is, everybody will have his or her own plate. We're going to seal it. We're going to stick it in the fridge. And all subsequent, many of subsequent experiments, you will select one colony and will try to find out what it is. Does that make sense? You will try to find out what the heck was that you isolated from, I don't know, bottom of your shoe. You can take two, for example. And one of the steps for this will be gram staining of the sample. So basically, first you add crystal violet to the cells, and it stains positive and negative cells equally. They all appear blue. You wash it away, the excessive crystal violet, and you add iodine, grams iodine, so-called mordant. So that mordant pretty much fixes the dye, okay? So the dye adheres to the cell walls. And cell walls much more prominent in gram-positive bacteria. Bacteria is still all blue, all remain blue. They then use alcohol to wash excessive dye away. What happens? That crystal violet dye does not really stick to gram-negative cells, so alcohol washes it off. It does stick to gram-positive cells. Despite of the alcohol, they remain blue. And then, the last step, you add safranin, which is red stain. So now your gram-positives look blue, gram-negatives look red. Does that make sense? It sounds like a lot of work. This stain can be completed in about 7 minutes. It is really, really simple. You have to be, the only thing, you have to be like careful and gentle. That's it. Nothing really fancy. If you can operate the pipette like this, you're good enough. That's maximum skills that you need. Okay. So these are some examples of differential stains. This is acid fast of Mycobacterium tuberculosis. Don't have to know about the mechanism. It works almost the same. The principle is the same as for gram stain, okay? The acid fast cells, they retain certain molecule after alcohol decolorization, okay? Capsule stain, India ink or Negrozin allows you to visualize capsules. You see those white, thin white layers around the cells? These are protective capsules. They prevent cells from being phagocytosed. So they're more resistant. It's actually a virulence factor. Some trait of the microbe that makes it more pathogenic. And obviously, when microbe can avoid your immune response, it is more likely to cause disease. Okay? This is endospore stain. Endospores are pretty interesting. So, endospores is like escape pods for bacteria. When it becomes really, really dry, and bacteria cannot survive on dry, some of the bacteria, called spore formers, protect their genetic material in the structure called bacterial endospore. Those cubbies on top of the cubbies and cabinets, if you would look there, you will find nothing but a bunch of dust. And nobody actually goes there, nobody wipes it off, nobody, there's no water, nobody touches it. So seemingly it's not really a hospitable environment for bacteria. But if you will take a sample from tops of the cabinets and plate it, you will see how those endospores that accumulate there in huge amounts will give rise to bacteria, to bacillus, different bacillus species. And you will see growth on the plate. So it's kind of a way to survive through the drought for bacteria. And you can stain for the endospores. You see those, the gray structures? It's endospores inside of the cells. And flagellar staining, that we talked about it, Bacillus cereus, making it thicker. This slides give you the summary of the methods. I'm not going to go over the same stuff again, but when you will study, when you will go over that at home, when you will prepare for exam or quiz, this can be useful because it kind of puts all of it in a very concise manner together. Okay? Does that make sense? So it gives everything that we talked about. You see almost the same illustrations. So before we will go to our samples, and before we'll probably do the quiz at the very end of the class. We're going to talk about this thing next. We're going to take a break, and we'll come back. We'll chat about the results.